Afternoon, folks. Hope everybody is doing well. I am here now to give you your second part in your lesson on mutation and cancer. If you have not already watched part one of this lesson, and for that matter, if you've not already watched my lessons on DNA and RNA, I strongly recommend you go back and do that prior to watching any further so that you have the necessary background for this lesson. If you are enrolled in my biology class, you should probably make sure that you have a copy of the handout from Blackboard on hand so that you have something to look at by way of examples and vocabulary and so forth for what I am about to tell you about. Now, in our last lesson, we defined the phenomenon of mutation as any change that occurs in the sequence of DNA. We identified a number of possible causes of mutations, such as radiation, viral interference, chemical interference, or so-called natural mutations. We also talked about types of mutation, ways in which mutation can affect the DNA, including a point mutation, an inversion, a deletion, an insertion, duplication error, all the various frame shifts, or a translocation error. We also then began talking about consequences of mutation for the gene itself. We had talked about the possibility of a back mutation that reverses a previous mutation. We talked about a missense mutation, which changes the amino acid sequence of the gene, or a nonsense mutation, which inserts a stop codon too early. For this lesson, I want to continue in that vein, looking at consequences of mutation, one of which is a disease that we call cancer. Now, at the end of the last lesson, I was outlining possible consequences of mutation for individual genes. And as I said a moment ago, we could have a missense mutation in which the mutation that we committed before, no matter what type it was, regardless of whether it was point mutation, inversion, or whatever, that my missense mutation means that the mutation we had caused a change in the codons. It changed the amino acid sequence that gene was coding for, and therefore caused a change in the proteins that the gene would produce. Now, you've got a possibility of a nonsense mutation. Nonsense mutation causes a stop codon to be inserted early in the gene, meaning that gene will be cut off before it is finished being transcribed. That whenever your cell tries to express that gene, it will only be able to partially build the protein because the stop codon will make it stop doing so early. We also have the possibility of a back mutation or reversion, which causes an earlier mutation to go away and basically sets things back to the way they were previously. One other thing I didn't mention here, and it's a really important one because it's a really common one, is that sometimes the effect on the gene will be no visible effect at all. We call that a silent mutation. In a silent mutation, we are going to be looking at a situation where the change made to the DNA does not actually change the amino acid sequence it is coding for. You might recall there is redundancy in the genetic code, right? That you can have more than one codon that represents the exact same amino acid. So if I have a mutation that changes a codon in such a way that it doesn't actually affect the amino acid sequence that it's coding for, no big deal. Technically, the gene has been mutated and changed, but the final product of the gene is not modified. By way of example, let's suppose that I have the codon ATT. It happens that the codons ATT, AAT, and ATA, all three of those code for the same amino acid. So if I have a point mutation that changes AAT to ATT, they both code for the same thing anyway. No big deal. I have not actually affected the amino acid sequence. Likewise, if I have an inversion that causes AAT to flip around and read ATA, not a problem. It didn't change the amino acid sequence any because both of those code for the same thing. Maybe you even have an inversion that flips around a larger number of nucleotides, but it still doesn't change anything. Maybe you've got an inversion that causes two pairs of nucleotides to switch places, except the sequence that was being inverted read GGGG. It's for the same thing in a row, and they flip places, it still reads GGGG, and therefore the resultant effect on the gene is nil. So a mutation does not always affect what the gene is going to do. Sometimes the mutation, because of redundancy in the genetic code or because of just flipping around things that made it look exactly the same as it did before, sometimes the mutation literally has no impact whatsoever on the protein product that that gene codes for. As a result of this, there are actually a couple of variants out there of common genes that basically have exactly the same effects. Like, it's conceivable that I may have a slightly different sequence for insulin in my DNA than you do, because I have a codon that reads ATA in my insulin sequence, and you have one in the same spot that reads ATT, and either way it codes for the same amino acid. So silent mutations are actually fairly common. And what I've just told you, by the way, is why redundancy in the genetic code is evolutionarily beneficial. Because since there are multiple codons present for any given gene, because I mean any given amino acid, that most amino acids have at least 2 codons that represent them, and sometimes as many as 6 or 7, that means that many minor mutations, mostly point mutations or inversions, don't actually change anything. The redundancy in the genetic code that way, in the amino acid code, is a defense against minor mutations. It's a way in which organisms basically can shield themselves against the smallest and therefore most likely possible errors in their DNA. Okay, so silent mutation, missense, nonsense, or back mutation. These are all possible consequences for any given gene that has experienced a mutation. Now let's go a little further out. Let's talk about consequences for you, the organism. Let's talk about consequences for what happens in your body specifically when a mutation has happened in one of your cells. Now, let's start with one thing. We got our science fiction scenario, right? You know, where like Peter Parker gets bitten by the radioactive spider and, you know, suddenly he's got spider powers, right? Well, for starters, no. But even assuming that you get bitten by the radioactive spider and it introduces radiation into your body and causes a mutation somewhere in your body, odds are pretty good that A, that mutation is not happening to all the cells in your body, and B, it's not going to be the same mutation even if it does happen to more than one cell. Mutation is a random event. Even if I introduce the same radiation to your body, it's not going to necessarily affect any given 2 cells the exact same way. Even if both of them have their DNA damaged, that radiation damage will not necessarily affect the same portion of the DNA or even the same chromosome. And therefore, I might end up being radiation exposed and have one cell over here that has a change in the 22nd chromosome and another cell over here that from that same radiation exposure has a change in the 18th chromosome. And lots of cells in between with no change whatsoever. Keep in mind that we have lots of defenses against mutation. We've got that nice thick skin around our bodies. Keep in mind that you have a plasma membrane and a nuclear envelope that any interference has to get through before it can even get to the DNA in the first place. And keep in mind that even if the DNA is damaged, we have proofreading functions and things like DNA Pol2 that can fix damage. So even if mutation does make it through to a bunch of cells at once, odds are pretty good that many of them will not, or sorry, maybe potential forces that can cause mutation afflict multiple cells at once. Many of them may not be affected. Or if they are affected, they might be able to be repaired. Or if they are repaired, it might end up being a silent mutation anyway. There's a reason mutation doesn't happen all that often, because we've got a lot of defenses. But we are interested here not in what happens if it doesn't happen. We're here to talk about what happens if it does. But keep in mind that we have to think about that at the individual cellular level. An entire organism isn't going to mutate because it got exposed to radiation. Individual cells within that organism might experience mutations. But we can't talk about the mutations of the whole critter. Now, it does matter which particular cells get mutated in terms of what happens next. You basically have 2 kinds of cells. You have somatic cells, which are the normal bodily cells, that affects, your skin, your bones, your muscles, all the different parts of you that are the operating parts of you. But we also have your gametes. We have your reproductive cells. That's where things get really interesting. Let's suppose we have a mutation that affects a gamete. It affects an egg or a sperm. Or maybe, let's say the mutation affects a germline cell, one of the cells present in the gonads that make eggs or sperm. Either way, the consequence of this is that whether it's affecting a germline cell, which then goes through meiosis, or whether it's affecting an egg or a sperm already produced, is that you can end up with a mutation in a reproductive cell. You can end up with a reproductive cell, an egg or a sperm, which, thanks to a mutational event, does not have the same genetic sequence that its mother cell had. You would have a sperm that has a gene that wasn't present in dad's body. Because let's be honest, what's happening here when you introduce a mutation, assuming it's not a silent mutation, what you've just done is you've introduced a new gene, haven't you? You've just introduced a novel possible gene located at that locus, that spot in the DNA. You added a new possible genetic variant to that cell. Now, odds of any given egg or sperm in your body actually being fertilized are very, very few. Even if you have dozens of kids in your lifetime, the overwhelming majority of your gametes will never, ever become part of a zygote. But let's suppose that our mutated sperm that we talked about a moment ago ends up fusing with an egg cell. And let's just assume that begins then developing and actually successfully implants in pregnancy, and we begin developing a child. Here's the thing. That zygote, all the genetic data that it had came from the egg and the sperm that made it. That zygote has no way of knowing that the mutation present in that sperm we talked about a moment ago was wrong. The zygote has no way of knowing, oh wait, that's a bad defective gene that's not supposed to be there. Because the only instructions the zygote has is the genetic information it received from the two gametes. In other words, that mutant gene that was produced in dad's body, or this could all happen in mom's body too, I just chose a sperm in the first example and followed it, but that mutant gene present in that gametes is now part of that organism from the moment of its conception. And because we all begin with that one zygote, that one fertilized egg containing DNA from mom's egg and dad's sperm, and that zygote then begins to undergo mitosis, and it becomes 2 cells, and then it becomes 4 cells, and then it becomes 8 cells, and becomes a bigger and bigger ball of cells, that means that that mutated gene is being copied in every last one of those cells. In other words, if I have a mutation that just hits random skin cells in my arm, a couple of my skin cells might be affected, but doesn't affect me as a whole. But if I have a mutation that hits my gametes or hits my germline cells, any individual conceived from that mutated gamete is going to have that gene present as part of their genome. It's going to be in every single cell of their body. It's just going to be a part of their DNA like any other gene. So in this way, mutations that affect a parent's gametes can end up causing new genes to appear in their children, even if that parent themselves does not have that new gene present throughout their body. So, we've conceived a kid with a mutated gene. What does this mean? Let's talk from most likely to least likely possibilities. The likeliest possibility is that it means nothing at all. Why? A couple of reasons. One, because of all the safeguards against changes in DNA they talked about a few minutes ago. The redundancy in the genetic code means that silent mutations are fairly likely. So, yeah, maybe I conceived a kid with a silent mutation that caused a slight change in the sequence of their gene for hemoglobin, but it changed the gene for hemoglobin from reading ACA to AAC, and those 2 codons represent the same thing. So that's one reason why no effect at all is a likely possibility. The other reason no effect at all is a likely possibility is because let's remember for a moment that your DNA is between 98 and 99% introns. In other words, the overwhelming majority of your DNA is not in fact coding for anything in the first place. By the numbers, between 98 and 99% of mutations that happen are going to hit a non-coding region of the DNA. Therefore, it's quite possible that you have mutations that are present throughout your introns, but I just mutated a non-coding sequence into another non-coding sequence. No big deal, right? Nothing changed. So there's a good possibility that any given mutation is simply not going to be an issue for you because it is not affecting a coding region of the DNA to begin with. And I should like to point out this is a big difference between prokaryotes and eukaryotes. Eukaryotes have lots of introns. It's normal for a eukaryote to have 98 to 99% of its genome not coding for anything. Prokaryotes have very few introns. A large percentage of prokaryotic DNA is actually coding exons. The majority of prokaryotes' DNA is coding regions. That's why they have so much fewer DNA, or so much less DNA, I should say, than eukaryotes, because they have a lot less introns, in addition to having less genes in the first place. Therefore, the odds that a prokaryote will actually be affected by a mutation are considerably higher than the odds that a eukaryote would be affected by the same mutation, because in the prokaryote, it's just much more likely that that mutation will actually hit one of its coding regions. Furthermore, the prokaryote, you might recall, doesn't have a nucleus to protect its DNA, and as a result, the DNA is more vulnerable and therefore more subject to outside influence. Those two factors together combined mean that prokaryotes experience way more frequent mutation than eukaryotes do, and that's a significant factor in prokaryotic evolution. So, odds are pretty good that the mutation that we just created will have no visible effect on the organism whatsoever, either because it was a silent mutation or because it hit an intron. Let's suppose it does affect a functional region of the gene. The most likely possibility there is that that gene fails. And in a developing organism, that means the likely option is that the organism will fail. That you basically are going to have a miscarriage in that. Well, I mean, not you personally, but you know that the result here is going to be a miscarriage, because if the fetus can't develop properly, it won't go through the whole process, you know. So oftentimes a mutated gene that deprives the developing fetus of necessary genetic information will simply result in the death of the child before they're even born. In fact, a majority of miscarriages, we believe, may be a consequence of mutation. So that's another possibility. Let's suppose you are born with the mutation. Let's suppose whatever the mutation was, it was something that you could survive to be born with. And again, we're talking about a mutation that actually hit your exons here. We're talking about a non-silent mutation at this point. A mutation that actually has a noticeable effect on the individual. What are the possibles here? First off, negative mutation. Negative or harmful mutation. You can call it either way. A negative or harmful mutation is going to have a deleterious effect on the individual that has it. In other words, you have the wrong instructions for making one of your body's proteins, and as a result, you have a health problem that is a consequence of not having the right protein. That mutation changed the instructions for one of your genes, and now it cannot properly produce the thing it's supposed to produce. Maybe that means that it makes a defective form of that thing. Maybe it means it doesn't make that protein product at all. It's like having grandma's old cake recipe, right? You know, you reach into the cupboard and you grab a random thing out of the cupboard and sprinkle it into grandma's cake. Odds are pretty good as picking out of all the options available in your kitchen that most of them are not things that are going to be good in a cake, and that you just added curry powder to your cake, or you just added mayonnaise to your cake, or something else terrible that's going to mess up your cake. We're the cake. So a negative mutation is by definition one that is harmful to the organism that has it. This might result in the organism having severe defects that cause it to die young. This might result in the organism having issues that it can survive with but cause it problems, but it carries them throughout its life. Maybe it has a shorter lifespan because of them. Or maybe it's a survivable issue but just uncomfortable. Now here's the thing about that. Once that negative mutation exists in an organism, as long as that organism can potentially reproduce, as long as that mutation has not either killed the organism before it got to reproductive age or rendered it reproductively unfit to begin with, then that mutation is just part of its DNA like any other gene that that organism has, and therefore that mutated gene can potentially be passed on to that organism's offspring, and therefore its offspring can have the same negative mutation that the parents did. This, folks, is the origin of what we call genetic disorders. When we talk about a genetic disorder is an inherited disorder. It's something where you got a gene for a problem from a parent that passed on that problem to you. Maybe the parent themselves didn't suffer from the problem because the gene might have been recessive, for example, but in you, it's being expressed, and now you have that issue. So anything that we think of as a genetic disorder, an inherited negative genetic condition, originated as a mutation. at some point in the past. But it's like I said before, that mutation only had to happen once. And as long as the individual with that mutant gene can reproduce and pass it on, it's now just an optional gene in the gene pool. It's now a variant that might be out there that you can mate with somebody who has it, and your kids can end up with it. Asthma, cystic fibrosis, spina bifida, a lot of allergies. There are a lot of genetic disorders out there, folks. There are a lot of inherited genetic conditions, and they range in severity from something that you can live with that's comparatively mild, like hay fever or asthma, to something that causes you significant problems in your life, like diabetes, to something that will kill you eventually if something else doesn't get you first, like Huntington's disease. So ultimately, mutation is the origin of every genetic disorder. But once that genetic disorder is circulating in the gene pool, it's out there, it's a variant gene that potentially can be inherited. That mutation does not have to happen a second time for you to end up with it. And odds are pretty good any genetic disorder you have was not because of a novel mutation in you. It's probably because of a mutation that happened dozens, hundreds, or thousands of generations ago. All right, let's take a look at another possibility. The next likeliest option, rather than negative or harmful mutation, is what we call a neutral mutation, sometimes referred to in evolutionary biology as selectively neutral. That is neutral in terms of natural selection, which is the mechanism of evolution. Now, when I say a mutation is neutral, I mean that it causes a visible change in the organism, but that change is neither beneficial nor harmful. It's just a variant. It doesn't actually mean anything for you as the individual. You work the same way. Like, okay, I have a mutation that widens my cheekbones slightly. Or I have a mutation that causes my hair to be a slightly darker color than it otherwise might have been. Or introduces a new hair color, Or I have a mutation that makes me a little bit taller or a little bit shorter than I otherwise might have been. Probably not a big deal, right? Odds are pretty good that as long as your vision works, a mutation that introduces a new eye color isn't going to affect your reproductive fitness one way or the other. So a neutral mutation is one that's just kind of there. It's neither beneficial nor inherently harmful. Neutral mutations are going to be the basic difference between you and everybody else you see around you. All the little cosmetic variations that we see in human beings in the structure of our bones, our facial features, our height, our skin color, our eye color, all the little tiny variants between one person and another that do not really impact your health or success originated at some point in evolutionary time as neutral mutations. And our population is just full of those things, and they tend to circulate kind of at random. Now, here's the cool one. Once in a while, if you reach into the cupboard and grab a random thing and sprinkle it into the recipe, you will improve on the cake. Once in a while, a mutation might make a gene work better. Doesn't happen often. A monkey with delicate genetic instructions, odds are not in favor of this being an improvement. But sometimes that little tweak changes a gene for the better. Maybe it causes a digestive enzyme to work more efficiently than it otherwise did so that you can get more nutrients out of a given food source. Maybe it causes a change in the structure of your tendons that makes you better able to transfer energy from muscle to bone and you can run faster. Maybe it causes an immune tweak in your body where your white blood cells are now more effective at fighting a particular disease and you have greater resistance to a pathogen. One way or another, it is possible, albeit rare, that a mutation will change the DNA for the better, that it will cause an improvement in the function of a gene rather than making it worse. That is what we call a beneficial or positive mutation. This is the rarest possibility of all for reasons that I have already outlined. But if you end up with a positive mutation, you got an advantage, right? You've now got a trait that's cool and useful and is helping you out in some fashion. Maybe it makes you better able to conceal yourself from predators because you've got a camouflage fur pattern, Or maybe it makes you better at finding food because your sense of smell is stronger. Or maybe it makes you resistant to a common disease that's present in your environment. One way or another, that positive mutation is helping you out. And what that means is that the organism with that positive mutation is more likely to succeed in its environment, and success translates to greater possibility of having offspring. And if you have more offspring than the next guy, but you've got the nifty gene, your kids probably have the nifty gene too. There's a greater likelihood that they will end up with that same advantage. And as long as the environment continues to be one in which that trait is still useful, your kids will have the same edge that you do, which means they will be more successful than individuals who didn't have it. They will probably have more offspring, and therefore that gene will become even more common in the next generation. The effect of this is that beneficial mutations tend to become more common in populations over time. That as long as that beneficial mutation remains useful in the environment where the organism is living, organisms that have it will do better than the ones that don't. Likewise, you can flip the converse of this, and you can see where a harmful mutation is going to reduce your odds of successful reproduction, right? I mean, if I have a genetic disorder that makes me more likely to die at a young age, odds are individuals with that disorder, averaged out over the population, will have fewer offspring than those that don't have that disorder, which means that harmful mutations will tend to become less common over time. They will tend to diminish in frequency over time because organisms with them are less likely to pass them on. This thing I have just described to you is the fundamental principle of evolution, folks. It is a thing called natural selection. Natural selection is often misunderstood. A lot of people characterize it as, oh, the strong destroy the weak, or the stupid weed themselves out of the gene pool, or, survival of the fittest, where only the strong survive. It's not how it works. It's a lot more complicated than that. The basic rule of natural selection fundamentally is organisms with more useful genes will have more offspring than organisms with less useful genes. So it's not even so much the strong beat the weak, it's really more this guy has an advantage that that guy does not. It doesn't have to be strength. That advantage might be you hide better. But one way or another, though, positive mutations are the engine of evolution. They are the thing that drives the evolutionary process forward because they are coughing up the variation that natural selection works with. Natural selection can only select among the available options. That's an important thing to know. Evolution is not responsive to the environment. There is nothing about me living in an Arctic environment that makes me more likely to get mutations that give me thicker fur. But if I happen to be the bear who got the mutation with the thicker fur, I'm going to do better in the Arctic environment, and therefore my cubs are going to have the thicker fur as well, and they're going to have that same advantage. So nothing about particular environments makes particular evolutionary changes happen. What natural selection is doing is it is selecting among the options that mutation has put forward. And whichever of those options are useful are the ones that are going to be more likely to be passed on. And if none of them are useful, that's the reason we have a thing called extinction. That no organism in the population had the right traits to respond to a given environment, and they all died off. One last thing I want to mention about possible outcome here. I talked about positive mutation, negative mutation, and neutral mutation. Let's talk about a thing called a nearly neutral mutation. A nearly neutral mutation is very, very, very slightly beneficial or very, very, very slightly harmful. That is, it's basically neutral, but it still causes maybe a minor problem for you under limited circumstances. My mother is allergic to monkeys. It's a fairly weird allergy to have. She only found out because she had a friend who had a pet monkey and it climbed on her and she broke out in hives. Odds are, you living here in, well, I guess I don't know where you're living watching this, but me living here in Pittsburgh, Pennsylvania, I have that inherited gene from my mother for a monkey allergy. It is highly unlikely to ever cause me a significant issue because how often am I going to be touching or coming into contact or even anywhere near a monkey? It might mean that, yeah, I can't get a pet monkey. It might mean that if I travel to an exotic location where there are some monkeys around, maybe I'm going to break out in hives. But in my day-to-day life here in Pittsburgh, monkey allergy is not technically a problem for me. This is a nearly neutral mutation. Likewise, or the mutation that caused it generations ago, whenever that happened, was a nearly neutral mutation. Likewise, it's possible that you have a positive trait that is only slightly or mildly beneficial. Not enough to really give you a significant advantage, but it's technically an advantage. Maybe I happen to have a gene that makes me resistant to dengue fever. Dengue fever is a tropical disease that is quite dangerous in certain parts of Central Africa. Living here in Pittsburgh, I will never be exposed to dengue fever in the wild. Yeah, if my life path happens to take me into the Congo and I get exposed to dengue fever, I'll be really happy to have that gene. But right now, in the environment that I live right now, it's not doing anything for me. That is technically beneficial, but not really helping me meaningfully. It even can be a thing that actually has an effect on your life, you know. Maybe I am like 2% more resistant to the coronavirus than other people. Yeah, I'm going to be grateful for that 2%, but odds are pretty good that it's, unless I get a lot of exposure, it's not going to make a big difference. Could make the difference, but tiny, tiny, tiny little edge, right? So that's nearly neutral. All right, so these are possible consequences for my offspring. If there's a mutation in my germline cells or in my gametes, I don't end up getting the benefits or drawbacks of that mutation. It doesn't affect me because the germline cells or the gametes are sending genes on to somebody new. But that mutated gene will then end up in all of the cells of my child, and my child, for better or for worse, will now have that gene as a new genetic variation in them, and they can potentially pass it on to their offspring. All genetic variation, positive, negative, or neutral, ultimately at some point in evolutionary time originated from mutational events. Okay, now that is what happens if the mutation affects my germline cells or my gametes. Let's suppose the mutation affects a somatic cell, an ordinary body cell, a bone cell or a skin cell, one of the many, many other types of 240 odd or so cells that make up the functional parts of my body. So I got a mutation in a given cell. Odds are pretty good it's not going to have much impact on me as a whole. I mean, if I got one cell out of the 100 trillion odd cells in my body that has a mutation, even if the mutation in that cell is causing that cell to be grossly dysfunctional, one cell isn't going to meaningfully impact how I work. So odds are pretty good that a mutation in any given cell in my body is not going to affect me on the whole one way or the other. But let's look at the individual cell level and see what it can mean for that one cell. The greatest likelihood here, as with previous things discussed, is no visible impact whatsoever. A cell gets a mutation. Individual cells have in their DNA a mutation, still 98% likely to affect an intron, and therefore no impact on the cell. Still, even if it does impact the exons of the cell, good chance of it being a silent mutation, in which case, no big deal. So this skin cell has a slightly different code for the protein keratin than the skin cell next to it. They both still make keratin, no problem. Let's suppose that the effect is beneficial. Maybe that one cell right there makes better keratin than the other cells around it. Okay, it's one cell. It's not really going to have an impact on me as an organism as a whole. Maybe even when it divides, now I have two cells that make the good keratin. Two cells out of how many millions of skin cells per square centimeter? Not a big deal. Let's suppose that it's a harmful mutation for that cell. Odds are pretty good that that cell is going to just die. You know, it's not able to carry out one of its functions properly, just like most harmful mutations that end up in our offspring kill them before birth. Most harmful mutations that occur in an individual cell cause that individual cell to die, in which case it's no longer an issue. If that individual cell is suffering a harmful mutation that does not kill it right away, sooner or later that cell is going to hit a checkpoint in the cell cycle. Remember the G1 and G2 checkpoints from way back in the day, you know, whenever we talked about cell division. That cell's going to hit one of those genetic checkpoints, and it's either going to be removed from the cell cycle and go into G0 and become quiescent for the rest of my life, or it's going to undergo apoptosis, in which case the cell self-destructs. Either way, it's not an issue for me, the organism. If the mutation is a neutral mutation or a nearly neutral mutation, same as a positive mutation, okay, this skin cell has a very slightly different variation than the skin cell next to it, but it doesn't affect its function in any way. No big deal. It doesn't matter if its DNA is very, very slightly different than the next cell over. Still doing its job. All right, so everything I'm saying here probably sounds like I'm saying to you, all right, so mutations that affect our normal somatic cells are not that big a deal, right? Not necessarily. First off, let's suppose that our mutagen is affecting lots of cells at once, and it's not one cell getting a harmful mutation. Maybe it's 1 million cells getting a harmful mutation because there was a toxin in my bloodstream or a large amount of radiation exposure, and a whole lot of my cells ended up dying because of mutations. We saw a very stark example of this in the aftermath of the atomic bombs that were dropped on Japan in 1945. Now, obviously, a lot of people died instantly from those titanic explosions, but in the weeks and months that followed, a lot of folks who didn't die when the bomb fell ended up dying of organ failure and a wide variety of problems that got lumped together under the heading radiation sickness. And what radiation sickness basically means is that lots and lots and lots of their cells, because they were bathed in terrible radiation, lots and lots of their cells ended up having mutations that made them stop working properly. And those cells either died or limped along in a dysfunctional way and weren't able to reproduce or underwent apoptosis. And those individuals ended up dying months or weeks or years later because so many of their cells had mutated and their bodies just couldn't work right anymore because they'd lost too many good cells. So that's one thing. But let's talk about one more possibility. Let us suppose that our mutated cell is defective. It's not working properly. But let us suppose that mutation is affecting it in such a way that it is now reproducing without stopping. That mutated cell is dividing and dividing and dividing and dividing again, much more rapidly than it ordinarily would. And now I have a mass of harmful mutated cells beginning to accumulate in my body. I've got this clump of cells that are not doing their job properly, but they're sucking up resources and they're continuing to grow and divide. Now I got a problem. That mass of abnormal cells that I have just described to you, you probably can guess, is called a tumor. T-U-M-O-R. What happens to it? A couple things can happen. First off, most of these cells that are mutated out of control and are growing out of control, most of them don't get very far. Most of them get stopped by our immune systems before they get a chance to divide at all. Our immune systems are really, really good at detecting damaged cells, noticing that they're damaged, and destroying them before they can become an issue. Furthermore, most damaged cells will self-destruct via apoptosis before they cause a significant problem. But if it survives, doesn't undergo apoptosis, our immune system's odds are pretty good they're going to get it anyway. We have entire populations of cells in our body called NK cells, natural killer cells, whose entire job is basically patrolling, looking for bad cells. And bad cells could be bacteria, bad cells could be parasites, bad cells can also be badly damaged somatic cells, which they can then gobble up and destroy. Phagocytosis. Now, let's suppose it doesn't get shut down. Or let's suppose that its rate of reproduction is outpacing the ability of the immune system to stop it. That little mass of cells can grow and grow and grow. Now, it's possible the body might be able to starve it out. It might be able to wall it off, basically denying it sufficient resources, encapsulating it so that it can't really grow anymore. Or it's possible that the cells in that mass might hit a point when they try to reproduce and they go into G0, that they don't pass a checkpoint and they just stop growing. And what you can end up with is just this lump of abnormal cells that aren't really doing much of anything. They're just sitting there. That is what we call a benign tumor. B-E-N-I-G-N. There's a silent G in there. B-E-N-I-G-N. Benign tumor. A benign tumor, you can live with it most of the time. Just because it personally is not doing anything sitting there does not mean it might not be a problem. For example, if you've got a benign tumor that is blocking up, a blood vessel or part of a digestive pathway or one of your pancreatic ducts or your gallbladder, that benign tumor can cause you some issues. Or maybe that benign tumor is pressing on a nerve and causing nerve problems as a consequence of being there. So a benign tumor could be problematic, and therefore you might want to remove it anyway. Odds are pretty good you have a benign tumor somewhere on your skin. Most of us do. We call them birthmarks. Birthmarks are benign skin tumors. A lot of moles are also benign skin tumors. They're just kind of hanging out. They're there. Not causing you any issues. By the way, also about 1 out of 10 of us have benign bone tumors. Benign bone tumors called osteomas are not at all unusual. They're little smooth masses of bone, kind of look like a little button. A lot of times they're in the eye socket, yeah, something like 1 out of 10 people have a small benign bone tumor. No big deal, just sitting there. But if you got that benign skin tumor, if you have that birthmark, you've probably been told this at some point in your life, keep an eye on it. Because if it ever begins to change, that could be indicative of a problem. Because that means that that benign tumor is no longer quiescent. That means that it's beginning to grow again. And let's suppose we have a tumor that's not so benign. Let's suppose we have one that's growing and expanding and adding more and more defective cells and sucking up more and more of the body's resources and getting bigger and bigger. What we are talking about here is a malignant tumor. M-A-L-I-G-N-A-N-T. Malignant. The word malignant means evil or vicious or, negative. And if you have a malignant tumor, that is the point when you have the condition that we know better as cancer. C-A-N-C-E-R. Okay, you all know about cancer. You've all lost somebody to cancer at some point. By the numbers, I'm sorry to report that some of you listening are probably going to die of cancer eventually. Cancer is a serious medical issue. We have an entire branch of medicine dedicated to its study and treatment, oncology. O-N-C-O, onco means having to do with cancer. By the way, if you also see the suffix oma, O-M-A, that refers to an abnormal growth. So like an osteoma. Osteo means bone, oma, abnormal. So abnormal bone growth. An osteoma is a bone tumor. Now, cancer is defined as the uncontrolled growth of mutated cells. That's what cancer means, folks. Cancer is the uncontrolled growth of mutated cells. Something happened, a mutation happened in a somatic cell. It is now dividing out of control. Dividing and dividing and dividing, and taking up the body's resources and interfering with its processes more and more. Now, here's the thing you need to understand. Cancer is not one disease. Cancer is an umbrella term for a whole collection of diseases, all of which have in common the unifying feature of uncontrolled cellular growth. No two cancers are exactly the same, and there will never be a cure for cancer. That's like saying, okay, will there one day be a cure for infection? Not all infections work the same way. Will there one day be a cure for viruses? Not all viruses work the same way. It's why we can't give you a flu shot and have it work on the coronavirus. There's never going to be a one-size-fits-all cure for cancer because not all cancers are the same. There are a tremendous range of possible mutations that can lead to cancers. And let me, by the way, disabuse you right now of a very common conspiracy theory in medicine, that there is some secret cancer cure out there that rich people have access to that you don't. There is no secret cancer cure. God, I wish there was a secret cancer cure, because then somebody could blow the whistle on it, but there isn't. And think about it for a moment. For there to be the secret cancer cure, that would mean thousands, if not millions, of physicians, medical researchers, technicians, business executives, pharmaceutical company representatives, and other people would all have to be on board with secretly hiding a cure that one day could probably save the lives of themselves or their family. And it would only take one of them to blow the whistle. So no, I'm sorry, there's no secret cancer cure. I wish there was. There's not. So what do we do about this thing called cancer? First off, it must be understood no two cancers are going to be the same. The mutation that caused my cancer is not going to be the same mutation that caused your cancer. Even if you and I have the same kind of cancer, the mutation that hit my lungs and gave me lung cancer will not look genetically the same as the mutation that hit your lungs and gave you lung cancer. As a consequence of this, some people's cancers go super fast, some people's cancers go super slow. You might have a cancer that's super aggressive and you get diagnosed in March and you're dead by November. You might have a cancer that you can live with for 20 years as it slowly grows before you have any harmful effects from it at all. The pace of cancer varies a lot depending on what kind of mutation you got. The severity depends on the location of the cancer. If I've got a little patch of mutated skin cells that I have a melanoma right there, that's skin cancer, that's okay. That's easy to cut off. I can just excise that bit of skin. Not a big deal. I've got a brain tumor inside my skull that's putting more and more pressure on my brain as it grows. That's an issue. If I've got a tumor that is growing in such a way that it's interfering with an organ, that's a problem. Blocking off an important duct, messing with the functions of an organ. If it's in a less problematic location, it may go unnoticed for a long time. Of course, one of the big dangers of any cancer is spreading, that it will not just stay in the part of the body where it began. Some tumors are what we call encapsulated tumors. An encapsulated tumor basically is all one mass growing all together. It's not extending into other nearby tissues. But a lot of tumors are not like that. Their growth is much more disorderly. They reach out. They just have little growths like tentacles penetrating the surrounding tissue. And a tumor like that, we always face the danger that portions of it are going to break loose, and cells can then spread to other parts of the body. This is especially a danger in any part of the body that has a rich blood supply. Bone marrow, your lymph nodes, your liver. Any situation where there's a lot of blood flow going through an organ means that a tumor that develops in that organ, you have the very real risk that cells will break loose from it and spread to other parts of the body. When that happens, we call that metastasis, 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 or we would say, you probably heard this term, the cancer has metastasized. The cancer has metastasized. That means it has begun to spread to other regions of the body. And once a cancer becomes metastatic, treating it becomes way harder. So, we've got this family of diseases. They all basically share in common uncontrolled cellular growth. Beyond that, they're highly variable. And that means that not only might the speed of the cancer vary between me and you, but the treatment might vary between me and you. Maybe a chemotherapy drug that works on me will have no impact on you whatsoever. Maybe your tumor is encapsulated and easily removed via surgery, and my tumor has all these tentacles reaching out into the surrounding tissue, and it's impossible to get them all loose. And even one new cancer cell can start the process over. Now, let me tell you an important thing that's kind of scary. You've had cancer. All of you listening to this have had cancer. You've probably had cancer this week, but your immune system shut it down before you knew. Because our immune systems are actually really good at shutting cancers down. Most cells have the ability to make a chemical known as an interferon, interferon. Interferons are basically chemical distress signals. They are sent up whenever a cell becomes badly compromised. Interferon chemicals have two effects. One, they warn nearby cells to prepare their defenses, and two, they send signals to immune cells that basically attract immune cells to their location. And interferon is basically a flag going up from the cancer cell saying, hey, I'm bad, come kill me. And so most of the time, cells that become cancerous begin cranking out interferons, and our white blood cells, things like our NK cells, show up and destroy them. So yeah, you've had cancer. Your body just shut it down before you had any idea you did. Sometimes our immune systems will catch up with a cancer. Sometimes you'll have cancer for a while and then its growth will just stop because your immune system has worked out a way to keep it in check, and it basically is keeping up with that cancer and starving it of resources or killing the cancer cells as fast as they come in. We say at that point the cancer is in remission. remission. And when the cancer is in remission, basically that means it's stopped growing. Sometimes we can induce remission through drug treatments that weaken the cancer enough that our body can basically take over and hold it back. The thing about remission is there's no predicting how long it will last. Remission can end at any time, and most folks who go into remission will find that eventually that remission ends and eventually the cancer returns. And that means you're going to have to keep an eye on it. Really, frankly, once you have a cancer at all, you're going to have to watch for that cancer for the rest of your life recurring again, because keep in mind, all it takes is one cancer cell left behind after any treatment that you did to get the whole thing started again. And maybe that one cancer cell goes quiescent for a while, once the body is walling it off and keeping up with it, but that doesn't mean it can't activate again. That's why you have to watch when your birthmark changes, because that means that previously benign tumor might have become active again. And we don't always know the reasons why that would happen. We do not understand remission. We don't know why sometimes cancers go into remission, and likewise, we do not know why they come out of remission. This is a big puzzle in immunology. What do we do about a cancer once you've got it? Assuming that your body did not shut it down adequately. How do we do anything about it? A couple of things we try. For starters, we will try to remove the tumor. That's a good idea right there. If you can localize the cancer and figure out where it is, and if it is in fact localized in an easily removable spot, cut the damn thing out. Operate on it, physically remove it. Sometimes that's all you need. Take out the mutated cells, you're good. Normally, we're going to want to take away a chunk of healthy tissue around the tumor just to make sure that we're not leaving cells behind within that nearby tissue. Obviously, some tumors are more complicated to operate on than others. If you've got a tumor inside your brain, it's probably not operable. But that's usually step one. And for some cancers, that's all you need. Cut off the little malignant melanoma on your skin, bam, skin cancer gone. Go about your business. Now, if that's not sufficient, next step is radiation therapy. We bombard the tumor and the area around it with radiation, gamma rays, that will kill dead just about any cell that are exposed to them. Now, normally, you're going to get kind of sick when you're under radiation therapy because we inevitably kill healthy cells along with the good ones. But in order to take out the cancer cell, that is usually a good idea, to work with that sickness for a little while as the trade-off. In fact, normally, whenever we remove a tumor, we'll couple that with radiation. We'll cut off the tumor, but we'll also do radiation to the tissue around it just to make sure we're not leaving behind stray cancer cells. If that fails, we go to chemotherapy. A lot of people use the words chemotherapy and radiation as if they meant the same thing. You may be undergoing both at the same time, but chemotherapy is drug therapy. Chemo, C-H-E-M-O, like chemical, Chemotherapy is basically attacking the cancer with a cocktail of drugs. There's no one single way of doing chemotherapy. There are lots of different chemo drugs because depending on where in the body your cancer is located and the particular way that cancer is manifesting based on the mutation that caused it, some chemo drugs will be more or less effective than others. So there's a wide range of chemotherapy options out there. I'm not going to go through all the different ways that chemotherapy can happen, but suffice to say, drug therapy to attack the cancer or at the very least to slow its growth enough that the body can take over from there. Now, a lot of chemo drugs have the downside of impairing the division of healthy cells even as they're impairing the division of cancer cells. So it's not unusual for you to get very sick while you're on chemotherapy. As I'm sure you're aware, people's hair falls out and stuff, you get sick to your stomach and have a hard time eating. You're in a lot of pain. This is not at all unusual when you are undergoing chemotherapy. And for some patients who are already in a delicate state of health, sometimes the treatment is worse than the disease. Some people are not eligible for chemotherapy because the effects of the drug itself would kill them. There are some more novel approaches being tried out to deal with different cancers. There's a lot of immunotherapy being tried with cancers right now. We're trying to develop vaccines against certain cancers. Some of you might be aware of Gardasil. Gardasil is a vaccine that you get against genital warts, HPV. Everybody should get it. It's a good idea, especially if you're sexually active. Even guys who don't normally manifest visible genital warts should probably get it because you might be spreading it to somebody that you don't know that you're giving it to. Here's the thing. The overwhelming majority of cervical cancers in women are caused by genital warts. Gardasil protects against 75% of the strains of HPV, including the main cancer-causing ones. So Gardasil, by blocking against that virus, is actually potentially blocking against a cancer. It's an actual cancer vaccine. We're trying other vaccine therapies that might look at actual cell surface identity markers on the surface of cancer cells, give you basically a vaccine against those, so your immune system learns to recognize them and then goes after the cancer cells more strongly. We're working on therapies to stimulate the production of interferons so that more cells go and attack the developing cancer. Immunotherapy, we call it. There are a whole range of novel cancer therapies being tried. One thing about this that's really cool, I told you that there's no one-size-fits-all cancer cure. But all cancers do have one thing in common. Namely, they all had to have a failure of apoptosis. Programmed cell death didn't happen. That basic cellular self-destruct mechanism that is supposed to take out a defective cell and prevent it from reproducing at the G2 checkpoint didn't happen. So the overwhelming majority of cancers, one way or another, needs to be blocking apoptosis, either because the gene that causes apoptosis itself was damaged, or because something else the cancer is doing is interfering with the process of apoptosis. But either way, apoptosis did not do its job for the cancer to take hold. So we're looking right now at basically ways in which cancers interfere with apoptosis. Because if we can find a way to block that, if we can find a way to stimulate apoptosis in cancer cells without stimulating it in healthy cells, or figure out what exactly is going on that made the apoptosis not happen to begin with, then we can potentially create a very broad spectrum cancer cure. Not even necessarily a universal one, because there's probably more than one way that cancers interfere with apoptosis, but it's a thing that's being explored. In the meantime, best thing you can do is prevention. And I'm sure you all know the basics here. You all know things not to do. You all know that diets rich in cholesterols and saturated fats are a precursor to developing a lot of cancers. You probably all know that a low fiber diet is a precursor to developing colon cancer. You probably all know that, of course, you all know that smoking is bad for you. You know, really, don't smoke. It stinks. You're going to have that nasty cough all the time. The amount of money you spend on cigarettes every single year would pay for a Disney World vacation every single year. You'd rather have Disney World than cancer, right? And if you say, I'm a smoker, gotta die of something sometime, I'm here to tell you, as someone who has watched 7 relatives die of smoking-related cancers, that's not the way you want to die. It is a horrible, slow, choking, gurgling, painful, agonizing death. You do not want to go through that, and you don't want to put your family through that. So there's basic stuff you can do. Exercise regularly. Regular healthy exercise stimulates your lymphatic system, which means that your immune cells are circulating better and getting more filtration through your lymph nodes, which takes out a lot of problems. Good diet, good exercise, avoiding smoking. There are a lot of things you can do to basically greatly reduce your likelihood of getting a cancer. Getting regular colonoscopies once you hit about your mid-40s or so that you're checking to make sure there are no colon polyps developing in a place where you don't see. Basic preventative maintenance medicine like that. Not all of us have the same odds. Some people have a genetic predisposition to develop certain cancers. The doctors have all asked you at some point, well, do any of your relatives have this particular cancer or that particular cancer? Because we have realized that some cancers tend to run in families. The reason for this is because of a group of genes called oncogenes. O-N-C-O-G-E-N-E-S, oncogenes. An oncogene, sometimes called a cancer gene, somewhat alarming name, basically is a gene that makes you more likely to develop a given cancer. An oncogene does not mean, okay, if I have this gene, I'm going to get this cancer. What an oncogene means is that under a given set of circumstances, I am more likely to develop that cancer than someone who does not have that oncogene. It is predisposing you to get a particular kind of cancer. The more we learn about the genetics of cancer, the more we are realizing that oncogenes seem to play a role in a significant number of cancers. It's possible that most cancers have oncogenetic precursors. I speak from my own family as a great example. My mom's side of the family, they all smoke like chimneys, and they all die of neck, throat, and lung cancer in their 60s. My dad's side of the family, they all smoke like chimneys, they all live into their mid-90s. Clearly, there's a genetic predisposition on my mom's side of the family to developing neck, throat, and lung cancers that my dad's side of the family obviously does not share. This is not to say, okay, if I have a family history of a given cancer, I'm going to get it. It is also not to say, okay, I have no family history of this given cancer, I'm not going to get it. It is to say, if you know that you've got that family history, here's what to watch out for. Here are the warning signs. Here are the precautions you can take. Do your best. You can take all the precautions in the world and still get the cancer. You can do all the bad things and still not get the cancer, but you might as well put the thumb on your scales and tip the odds in your favor. And increasingly, genetics are going to be a part of how we treat cancer. Increasingly, we're making profiles of the genes of cancer cells and figuring out what mutations are there so we can figure out targeted ways to go after them. Gene therapy for cancer is a realistic thing we're going to be doing in the next couple of years. We're already doing gene profiling on cancer patients in order to figure out, okay, here is the way your particular cancer works, and therefore we can tailor the treatment to best fit the particular mutation that's afflicting you. In the future of cancer therapy, every cancer patient is going to get a genetic profile done, and we're going to look at them and say, okay, here are the oncogenes that you might or might not have. Here is the likely kind of mutation responsible for your cancer, so we can tailor make a therapy better suited to your particular problem that we know with that mutation, this drug will be better, or we say, okay, you've got that oncogene, you can watch out for the warning signs of this particular kind of cancer. This is kind of where we're looking at. There's no one-size-fits-all cure. There's no easy fix. But that gives you the basics. I've run a little bit longer than I normally run with one of these lessons, but I felt like it was important to tie this off. I thought it was an important discussion to have. I hope you all have a good rest of your day. Stay healthy and safe, folks. And by the way, one last thing. If any of you liked the evolution stuff that I was talking about in here, I do an entire course on basically adaptation and evolution. A lot of my biology lectures and my Bio 2 class deal specifically with that topic. I'm an evolutionary biologist, paleoanthropologist by training, so evolution is kind of my jam. And if you want to know more about that, just let me know. All right, y'all have a great afternoon.